Hi, everyone. The concept of reversing age-related decline, even within a single organ, remains a fundamental pursuit in biology. Today, we're going to look at a recent study by Kothmeyer and colleagues that explores this question within the intestine. We'll be examining how the turquoise killifish is being used as a model system to understand intestinal aging, and how a dietary intervention, specifically intermittent fasting, may affect this process.
So here's how we'll walk through the research. We'll begin by introducing the killifish model and its particular intestinal anatomy. From there, we'll quantify the structural and cellular decline that occurs with age. Then we'll look at the experimental setup where an intermittent fasting regimen was introduced. After that, we'll zoom in on the molecular data to understand the mechanisms of rejuvenation, and we'll conclude by discussing the broader scientific implications of these findings.
All right, let's start with the model organism itself. The work is centered on the turquoise killifish, Nothobranchius furzeri, an organism whose biology makes it a particularly useful system for studying the full spectrum of aging in a vertebrate.
The reason this fish is so valuable for this kind of research is its lifespan. It is the vertebrate with the shortest known lifespan, completing its entire life cycle in about 20 weeks. Crucially, during this compressed time frame, it experiences degenerative processes that are analogous to those observed in mammals. This allows researchers to conduct longitudinal studies of the aging process in a way that just isn't feasible with longer-lived organisms.
Now, one of the most distinct anatomical features of the killifish gut is that it lacks a stomach. It's described as agastric, which is supported by the absence of genes for pepsinogen or the proton pumps required for acid production. Instead of the typical mammalian villi crypts, its intestine is structured with analogous folds and interfold regions, or IFRs. It also lacks Paneth cells, which are key components of the mammalian stem cell niche.
So this raises a question. How does a stomachless fish handle ingested food? To investigate this, the researchers measured the gut's physical properties using brilliant light scattering microspectroscopy, which assesses elasticity. They found that the anterior portion of the intestine, known as the intestinal bulb, is significantly more elastic than the middle and posterior sections. This suggests a functional adaptation. Without a stomach, this flexible anterior section likely serves as the primary food receptacle.
Okay, so we have a good picture of the system. Now, let's examine what happens as it ages. In this next section, we'll review the specific ways the killifish gut declines, looking at the evidence from both a macroscopic and a cellular perspective.
The study was quite methodical, sampling fish at 4 key life stages. These were selected to parallel human development, moving from a larval stage analogous to a neonate through adolescence and adulthood, and finally to old age at 18 to 20 weeks. This careful staging is what helps make the findings translatable to mammalian systems. One of the first clear signs of aging they documented was a physiological decoupling. As the fish aged and their body length continued to increase, their intestines actually began to shrink. The intestinal length, diameter, and total surface area all showed a significant decrease after the fish reached adulthood, pointing to a progressive loss of absorptive capacity. This observation of the gut actually shrinking, well, it leads to a critical question. What is happening at the cellular level to drive this structural change? The investigation logically turned to the engine of gut renewal, the intestinal stem cells.
Similar to the mammalian gut, the killifish intestinal epithelium undergoes constant self-renewal, a process that originates with intestinal stem cells, or ISCs. The study confirmed that these ISCs are located in clusters within the interfold regions. From these IFRs, new cells proliferate and migrate up the intestinal folds and are eventually shed at the apex. It's a continuous cycle that maintains tissue integrity.
To measure the pace of this renewal, the researchers used a double labeling protocol to track cell division. They calculated that in a healthy 12-week-old adult fish, an intestinal stem cell completes its cycle in approximately 50 hours. This provides an important baseline for what's to come.
When they performed the same measurement in old, 20-week fish, the cycle time increased substantially, to approximately 76 hours. This represents a significant slowdown in the gut's regenerative capacity. It's a quantitative marker of stem cell exhaustion that correlates directly with the macroscopic shrinkage of the intestine.
So we've documented this age-related decline. The gut shrinks and its stem cells slow down. This brings us to the intervention phase of the study, where the researchers introduced intermittent fasting to see if this process could be modified at a molecular level. To understand the impact of fasting at a molecular level, the team performed a transcriptomic analysis. This table summarizes the number of differentially expressed genes between groups.
Now, if you look at the comparison between adult and old fish on a standard ad libitum diet, you see 282 genes change. But let's focus on that bottom row. When you compare old fish on that standard diet to old fish that were intermittently fasting, you see 569 genes with altered expression. This is a very substantial molecular response to the intervention.
And this leads to the core finding from the transcriptomics. The gene expression profile of an old fish that had undergone intermittent fasting was far more similar to that of a young adult fish than it was to an old fish on a standard diet. This is a clear molecular signature of rejuvenation.
So if the gene expression profile is reverting to a more youthful state, what are the actual mechanisms at play? In this next section, we'll dig into the specific genes and cellular pathways that were affected to better understand how this happens.
One of the first significant findings was related to the peripheral circadian clock. As you can see, in the old fish fed normally, the expression of core clock genes like PER1 and PER3 is significantly downregulated. However, in the fish that underwent intermittent fasting, the expression of these same genes was maintained at levels comparable to those of young adults, which suggests that the dietary regimen helps preserve circadian function in the aging gut.
We see a very similar pattern when we look at genes involved in nutrient metabolism. For example, genes involved in lipid transport, like ApoA1B, which normally decline with age, were maintained at youthful expression levels in the fasting group. Conversely, genes like CTSL1 that are upregulated with age were brought back down toward the adult baseline. Essentially, fasting appeared to help the gut maintain its youthful metabolic program.
And this is perhaps the most direct evidence. When they analyzed genes related to the intestinal stem cells, they found that key markers for ISE maintenance and function, genes like AK2, HOPS, TERT, and SOX9B, were all downregulated in the normally fed old fish. And again, intermittent fasting reversed this trend, restoring their expression to levels seen in adult animals. This provides a molecular link between the diet and the health of the stem cell population.
So, what do we make of all this? In our final section, let's synthesize these findings and consider the bigger picture. The data from this study led the authors to propose an interesting model for how intestinal stem cells are maintained in this system, one that may differ from established mammalian paradigms.
The model they propose is this. It may be that in the killifish, the pool of intestinal stem cells isn't a fixed, dedicated population. Instead, they suggest that other cells, such as progenitors or possibly even some differentiated cells, can be dynamically recruited to become stem cells when needed. The idea is that signals from the surrounding niche can instruct a nearby cell to adopt an ISE fate. This high degree of cellular plasticity might represent a more ancient, flexible mechanism for tissue regeneration.
And even if the precise stem cell biology differs, the effect of the dietary intervention appears conserved. As the authors themselves state, the maintenance of adult gene expression in aged killfish by IF, and presumably the maintenance of ISE function, is consistent with the findings of studies conducted in mammals. This reinforces the value of the killifish for uncovering fundamental mechanisms of aging that are broadly applicable.
In the end, this research connects three fundamental biological processes: nutrient sensing, circadian rhythm, and stem cell maintenance. It provides strong evidence for an evolutionarily conserved link between what an organism eats, its internal clocks, and its capacity for tissue regeneration. The finding that an intervention like intermittent fasting can positively influence all three domains in an aging vertebrate opens up compelling new lines of inquiry for understanding our own aging process.